Hello, welcome to this new webinar hosted by Bionano Genomics entitled Known Unknowns, Unraveling Genomic Inversion and Their Role in Disease Traits. I'm William O'Bannon with Bionano, and I'll be your moderator for today's presentation. Anyone interested in genomic structure for the sake of understanding genomic structure should be interested in today's talk. Inversions are a class of structural variation in the DNA that are the most difficult to detect. And to make matters worse, they're often flanked by repetitive sequences. Those people studying inversions are considered the superheroes of genomics researchers. And Dr. Chris Grochowski of the Baylor College of Medicine has chosen this extremely complicated subject as his specific field of study. He does so under the care and guidance of well-respected leaders in their fields, Drs. Claudia Carvalho and James Lupsky. And before we begin, I would like to remind the audience, as always, to stick around for the live Q&A session with Chris, which will begin directly following his presentation. Don't be shy. Feel free to send in your questions at any time during the presentation. We'll try to get to as many of your questions as possible. If you do not see a Q&A window, at the bottom of your screen, you will notice a purple Q&A box in the control panel. Click that box and a new object for asking questions will appear in your console. All other window objects within the attendee console, such as the presentation slides and the live view window, may be adjusted in size and location to suit your individual viewing preferences. Okay. Now it is my great privilege to turn the floor over to Dr. Chris Grochowski.
Thank you so much, Will, for that introduction. And again, my name is Chris Grochowski. So I'm a PhD candidate in the Department of Molecular and Human Genetics here at Baylor College of Medicine in Houston, Texas. And today I'll be talking about our research in trying to understand genomic inversions and their role in disease traits. So genomic inversions fall under a class of genomic aberrations known as structural variations. So here I just have a couple of examples showing a copy number loss, where a genomic fragment is lost, copy number gain, where a genomic fragment is duplicated, and an inversion, where a genomic fragment is inverted 180 degrees. There are also several other types of structural variations, such as translocations and insertions. So this kind of shows the most simple type of structural variation, and if they were all this simple, I would have nothing to work on for a PhD. But lucky or unlucky for us, these structural variants can have very complex features. So this is just a quick figure from a review article from Claudia Carvalho that shows kind of these complex structures that can be found, where it's not just a normal duplication, but may be a duplication followed by a normal copy number and then another duplication, or duplications and triplications that are inverted, rolling circle type mechanisms. There are tons of different variants of structural variations and very complex features that we have to study. And for the sake of this talk, I'll be kind of focusing more on the inversion states that we find in these complex cases. So inversions, again, are a type of structural variation. They can be found in a paracentric fashion, where we have two breakpoints that occur on one chromosomal arm and a 180-degree turn of DNA. They can also occur in a pericentric fashion, where we have breakpoints that occur on either chromosomal arm, and a 180-degree turn, including the centromere, occurs on that chromosome. So inversions are important to study for several different reasons. Not only do they have evolutionary significance, this paper highlighting their role in creating butterfly mimicry, they're also very important for the evolution of new genes as well as speciation. They also have human pathogenic consequence, here highlighting their role in hemophilia A. And in fact, this paper highlights that 42% of severe hemophilia A is caused by a common inversion event, as well as their mechanistic origins of how, where, and why in the genome these inversion events are occurring. And in fact, I'll go over this a little bit later, but this duplication-triplication structure that's inverted is actually one of the 12 most prevalent mutational signatures that's also present in cancer genomes, just kind of highlighting the importance of studying structural variants, not only in the constitutional genome, but also in the cancer genome. So even though inversions are very important to study, they're very difficult to study, as Will was kind of leading to in the beginning. So normally inversions were originally detected by traditional karyotyping analysis, and they have that here seeing the G banding structure. But again, karyotyping only has the resolution of about 5 to 10 megabase. And so if we have inversion events that are smaller than that, we won't actually be able to see it on a karyotype. Next is kind of the gold standard of studying structural variants is an array comparative genomic hybridization. But since many inversion events are copy number neutral, if we looked at an array CGH, it would look like a totally copy number neutral event, and there would be no obvious indication that there's an inversion that's occurring. And finally, when we try to use things like whole genome sequencing and short read paired end sequencing, these inversion events can occur within repetitive elements that are kind of invisible to normal short read sequencing technologies, making it that much more difficult to kind of study these structural aberrations. So for that, we kind of wanted to move on to new technologies that would give us an ability to view the genome in ways that weren't previously available to us. And one of those technologies that we've really had some nice success with is optical genome mapping. And so just to give a brief overview, so we're not actually sequencing the nucleotides, but we're actually taking ultra-high molecular weight DNA. So it's very, very, very long lengths of DNA. And then we're sequencing, though, or we're applying specific DNA labels to recognition sites and tagging the specific recognition sites of those very long DNA molecules. So from there, the DNA molecules are basically applied to an electronic or an electric flow. They go through these pillars to linearize so that we're able to take pictures of all of those fluorescent tags that are occurring on the fluorescently tagged DNA. And just to kind of show a video that I think is just really cool to look at. So here we are seeing, once it starts playing, basically the DNA molecules. So you see the green, these DNA molecules are flowing through those pillars and actually going into those nanochannels. And so those are individual strands of DNA that are flowing in those nanochannels that are fluorescently tagged. And then a camera is able to take a picture of the DNA. And you can kind of see on the right, that blue image that has those beads on a string type structure. So all those fluorescent tags that are labeled. And so what we can do there is that we can take computational software that will look at all of those images, take out where the molecules are and where all of those sequence motifs are, and then build together a de novo sequence of our genome assembly that we can then compare to an in silico reference to try to call structural variants that are in an individual genome. So why are we using this technology? So the gold standard and what we kind of really learn structural variants on is this array comparative genomic hybridization that I have an example of here. So here we can see an array that looks at a region of the genome on the X chromosome that targets a gene called MECP2, which I'll talk about a little bit later. So we can see a classic structure that goes from a normal copy number to a duplication to a triplication, back down to a duplication, and back down to copy number normal. And so one of the problems with an array CGH is that although it's very precise in telling us the copy number state of where it occurs, we have no information of how those genomic fragments are actually positioned on a linearized chromosome. And so we kind of have to move on to technology that can help us understand the architecture in a way that previous technologies just didn't give us that information. And so not only does it not tell us the architecture, but it also has problems in that there are repetitive sequences of the genome that things like array CGH and things like whole genome sequencing just can't target. And so here I'm showing that same MECP2 region, and within MECP2, there's actually a gene cluster known as the opsin cluster. That's 3 gene repeats that are highly similar. And so on an array, we're not actually able to target probes to that region because we have no information of how they're different. If we look at that same region in whole genome sequencing, we can also see that we have no information because basically the reads can't map to a non-unique sequence. And so we really have to move on to other technologies that allow us to see through these regions and to see the architecture that's at a given genetic loci. And again, here I just want to show, and I'll highlight this a little bit later, but when we look at things like optical mapping, this is looking at that same region that shows MECP2, and we actually have single DNA molecules that span right through that opsin gene repeat cluster. So it really allows us to see the entire region on a single DNA molecule. And so when we study things like structural variants, we really want to study them in what's called in cis, so on the same DNA molecule. And so it's very, very, very important for us to understand how that structural variant looks on the same DNA molecule versus other technologies and other methods that basically shred the DNA and then try to piece it back together. So it's nice that things like optical mapping allows us to see these aberrations on single DNA molecules. And this one I think was really cool because it highlights a DNA molecule that spans almost 800,000 bases as a single DNA molecule right through this entire region. And so today I kind of want to go through a few inversion cohorts that we've been working on. The first one looking at a multiple de novo inversion case on a single chromosome, and the second one looking at complex genomic rearrangements involving the MECP2 locus. So to start out with that multiple de novo inversion on a single chromosome, this was a really wonderful collaboration that we had with scientists and clinicians at the University of Sao Paulo in Brazil, as well as the Karolinska Institute in Sweden. And this involved a young girl who was 4 years old who presented with hypotomia, strabismus, congenital heart defects, as well as some craniofacial dimorphisms. You can see, kind of appreciate that here. She also, so we see the pedigree here with her unaffected parents as well as one unaffected sister. And so her previous genetic makeup showed an adjuvant whole genome array that had 180K probes, again, genome-wide, and that showed as totally normal. So there were no aberrations that were found on that array. Traditional karyotyping, though, did show what appeared to be two balanced inversions, both a pericentric and paracentric inversion that seemed to be localized on chromosome 6, as detected by G-banding. And so what it looked like is that this inversion event could disrupt the gene ARID1B. And again, these events were then confirmed through fluorescent in situ hybridization. So ARID1B is a gene that has a role in chromatin remodeling and is also causes the autosomal dominant disorder Coffin-Siris Syndrome. And so that disorder is characterized by developmental delay, coarse facial features, sparse scalp hair, and notably absence of the fifth finger or toenail. So again, that was actually one feature that wasn't present in this individual. And so they really weren't sure as clinicians if this was actually a disruptant of ARID1B or if there were other genetic factors that could play into her clinical phenotype. So again, this is just kind of showing that original FISH analysis that was done, and we could see that from the FISH analysis that there was a pericentric and paracentric inversion that was occurring on this individual. And so if we bring together the initially proposed architecture, we can see about a 90 megabase structure, or a 90 megabase pericentric inversion that occurs with the centromere, as well as a 47 megabase inversion event that was also occurring. So what we really wanted to do is to kind of apply more in-depth methods than just karyotyping in FISH to see, can we really tease out the structure, and are there other complexities that weren't apparent from that original analysis? So taking that original karyotyping in FISH, we then applied Trio custom high-resolution array CGH. So this looked at a very high resolution that looked at less than 1,000 base pairs per probe that gave us a really in-depth view of chromosome 6. We also applied Trio whole genome sequencing, as well as Trio genome optical mapping. And so again, our questions really were, are there other genomic complexities that are not fully appreciated on the original array CGH and karyotyping analysis, as well as can we really confirm that ARID1B is disrupted, causing the phenotype, and really the phenotype and symptoms of this patient. So we begin by looking at the array CGH. So again, that array CGH shows the mother, father, and proband, and what we look at is basically looking at the short arm and long arm of chromosome 6. And from this, it's totally clear. So even looking at a very high resolution array, we can tell that there are no structural variants, or no copy number variants, that is, on this chromosome 6. And we even zoomed into ARID1B to really make sure there was nothing small we were missing. And it looked like ARID1B, there were no copy number variants as detected by the array. And so when we look at things like whole genome sequencing, we actually go to those points that we believe the breakpoints are occurring, and we do see what's known as these soft clip reads. And so again, one side is connecting to reference, and the other side that kind of has that rainbow is connecting to the other side of the breakpoint. And so we have those proposed 3 junctions that occur here in the whole genome sequencing data. And what we can do is actually look at that junction and go to see where it's mapping to. And so when we did that, we actually found a more complex structure. And so this is where we started to get kind of confused, because it looked like there wasn't just that pericentric inversion and paracentric inversion that was occurring, but there were actually more breakpoints on this chromosome than could be explained by just those 2 structures. And so instead of the 3 breakpoint junctions in the proposed architecture, we in fact had 6 breakpoint junctions that were kind of apparent. And so if we look at those 6 breakpoint junctions here, we can see that there are then breakpoints that are occurring in the whole genome sequencing data that are connecting to different portions of this chromosome in a way that we weren't really sure how to connect the structure back together. So for that, we really want to apply optical genome mapping to say, can we resolve this chromosomal architecture and really understand the true structure of this chromosome 6. So for that, we kind of can see, if we just look at the Circos plot, those 3 junctions that form that large pericentric and pericentric inversion event. So that can be easily visualized on the Circos plot here. And those are, again, the inversions that are larger than 5 megabase and can be viewed on this Circos plot. But again, we know that there are more junctions apparent, so we really wanted to dive in to say, can we see those junctions? And so what was really cool is that when we started to go in and really look at these exact genomic regions, we could go into the optical mapping data and say, okay, we know that there's a breakpoint occurring at this genomic location. What is that genomic location connected to, and in what orientation is it connected to? And so here I just kind of have a view of our optical mapping data that we can see. We know a breakpoint occurs at this 16 megabase region, and then we can see in an inverted orientation, it's connected to this 11 megabase region that's, again, on the long arm of chromosome 6. So this is just going to show a quick video of kind of how we visualize this data in our optical mapping. And what we see here is basically we can look at each of those sequence motifs and see at what genomic location they are. And we can see as we kind of scroll our mouse across that we're moving in an upward orientation. And then when we hit that inversion event, we start going in a negative orientation to the Q-arm connection for this breakpoint junction. And I think what's really helpful for us when we're looking at optical mapping data is that we can actually look at the raw molecules and see that there's a breakpoint junction not only occurring, but we can see the molecule support for that breakpoint junction that's connecting those 2 genomic locations together on individual DNA molecules. And so again, this is kind of another visualization that we have, this one looking at junction 2. And so what we really can go through is start to really look at these junctions, to look at each breakpoint that we visualize and see not only where is it connecting in the DNA, but in what orientation is it connecting in the DNA. So we can kind of really go through each of these points, and we know basically where they're connecting. We can go to that genomic location in the optical mapping data, and we can really accurately see where it's connecting to and in what orientation it's connecting to. And I really wanted to highlight this junction 3, because this is actually the junction that disrupts the gene ARID1B. And so we can see here, ARID1B is connecting to reference to a point that's about in the exact center of the gene, and then it's connecting to a different point in the reference. So this is the point in this individual that we know genomically based off of optical mapping and other data that ARID1B is being ripped apart and connected to 2 different genomic locations on chromosome 6. And again, we have junction 4 here showing a connection to the 158 megabase region, connecting to an inverted orientation to that 160 megabase region. And so I think what's really nice to visualize data in this fashion, because we can actually see that an inversion is occurring very logically, right? So I don't have to kind of think about exactly how the structure comes in mate pairs, but I can see in the optical mapping data that an inversion is occurring because the sequence motifs are literally in an opposite orientation. So this is just kind of a view that we are kind of looking at the optical mapping data. And I like showing this view because I think it's hard sometimes to understand what I mean by when we see, but we can see here, I scroll my mouse across, I can see the genomic location of each of those sequence motifs, and I can see exactly where it's connecting to genomically on that chromosome 6. And again, we have junction 5 here. So again, this is connecting those 2 genomic locations in an inverted orientation. So we have a break that's occurring in 158 megabase going forward, or in a positive orientation connecting to a negative orientation of 157 megabase location on this chromosomal arm. We can see here, I think if you just look at the sequence motifs, it's very obvious to a viewer that an inversion event is occurring at this location. And kind of just the last junction here we have showing 160 megabase connecting in a positive orientation to a negative orientation at 159 megabase for this chromosome 6. And so when we bring together all of that data and we really look at it as a whole, we can start to resolve this chromosomal architecture. And so it's not just that one pericentric and one paracentric inversion, but But looking at this data together and really looking at the orientation of each genomic fragment and where those breakpoints are occurring, we can see that it's actually 1 pericentric and 3 paracentric inversions that are all occurring on this chromosome 6. And really interestingly, all of those inversion events are kind of connecting with each other, which is really, really interesting and to study. And so I kind of put up here, we can see the location that ARID1B is being ripped apart and connected to 2 different genomic locations on this chromosome. And so using our data that we have for the orientation from optical mapping, as well as our whole genome sequencing data, we can actually get the nucleotide level resolution of every breakpoint junction that's occurring and look at features that are occurring at those breakpoint junctions. And again, this is just kind of showing our visualization of all of the methods that we can apply to really tease out the true structure of this chromosome, including whole genome sequencing, optical mapping, we can form a junction alignment, and we can finally do Sanger validation of that breakpoint junction to prove that breakpoint junction is occurring in the orientation that we would suspect. This is just kind of a final figure showing our architecture. And again, I showed the gene ARID1B up here, showing exons 1 through 19. And that breakpoint is occurring between exon 4 and 5. And so what I think is really interesting is that this breakpoint is not occurring in an exon and is totally copy number neutral. So if you were to look at an array CGH like we did, it looks totally normal. But equally, if you were to look at an exome sequence, this would also look totally normal because there is no change in those coding sequences that are being sequenced in an exome sequencing reaction. And so what's actually happening is that this structural variant, this inversion, is ripping the gene apart and connecting it to 2 different locations. So this complex structure kind of looks like a chromotriptych-like event, so kind of a chromosomal shattering. And since we're able to look at the junction features, we can see that non-homologous end joining looks like it's the likely formation mechanism for these junctions. And again, the 95 megabase and 46 megabase inversion could be resolved by karyotyping in FISH, but it took other methods such as optical mapping and whole genome sequencing to really resolve the true structure and to find those secondary or additional inversion events that were occurring in this individual. And finally, ARID1B, we could prove through many methods now, was disrupted in this patient, causing their Coffin-Siris-like syndrome. So that was kind of our multiple de novo case in a single chromosome, but now I kind of want to move on to a more complex region, and that's rearrangements involving the MECP2 locus. So MECP2 duplication syndrome is a neurodevelopmental X-linked genomic disorder that's caused by copy number gains at the very end of the long arm of chromosome X in males. And patients have a variable clinical presentation and severity that kind of runs the spectrum. And I just have a picture of a few patients from a paper here. But when we think of MECP2 duplication syndrome, so this is a slide that I had showed previously, it's really a molecular diagnosis, right? So when we look at an array CGH, it's a copy number gain in MECP2 is what we know causes MECP2 duplication syndrome. And so this just kind of shows an array CGH at that location that shows a copy number neutral going to a duplication, in this case going to a triplication of MECP2, down to a duplication, and down to copy number normal. And so we really want to try to tease out the structure and the architecture of this genomic location to really understand truly what's happening in a linearized chromosome. And so, lucky for us, this is a very well-studied sample cohort within the laboratory, and it's been studied for many years now by Claudia and others. And we now have a large breadth of samples that we have to actually look at genomically. So they kind of span from a simple duplication, terminal duplications, translocations, as well as more complex cases involving dupe, normal dupe, dupe, trip, dupe, and recombinant chromosomes. And so what I really want to focus on for these cases are these dupe, normal dupe, dupe, trip, dupe, and these recombinant chromosomes, because these are actually cases where genomic fragments can be found in an inversion state. And so this duplication-triplication inverted duplication structure was really pioneered by Claudia Carvalho in the lab. This kind of just highlighting her Nature Genetics paper that came out in 2011 that kind of teased out that structure of how it comes together and kind of what genomic features at that locus causes this structure to occur. And recently, this has been highlighted in this Pan-Cancer Genomics Consortium, which is another landmark paper that really shows the structure of structural variants that can be found in cancer genomes. And astonishingly, they found this same dupe-trip inverted dupe structure was found not only in cancer genomes, but also we find in the constitutional genome. And in fact, they showed is one of the 12 most prevalent mutational features that are present in cancer. And kind of what I was saying earlier, but this really hammers home the point that these structural variants are recurring biological mechanisms that are occurring both in cancer and the constitutional genome. And truly studying them and understanding their structure and their true structure is really important, not only for cancer research, but also constitutional genome research. And so when we look at this structure, so again, at the very top, I have an array comparative genomic hybridization that shows that copy number neutral going to a duplication, triplication, back down to a duplication, and back down to copy number neutral, with MECP2 kind of sitting right in the middle of that triplication. So if we look at this in whole genome sequencing, that junction 2, which is basically the beginning of the triplication connecting to the beginning of the duplication, that occurs within unique sequence. And so that we can actually see in whole genome sequencing. We can visualize those soft clip reads, and those are reads that one side is connecting to reference, the other side is connecting to the other side of the breakpoint. We can form a junction breakpoint sequence, and we can basically resolve that junction. But the problem that we run into is here at junction 1. And junction 1 is a problem because that breakpoint is actually occurring within low copy repeats and segmental duplications. And so that's a region that we don't have information in our whole genome sequencing data that actually tells us that these 2 points of DNA are connected together to form this structure. And so what we really wanted to do was apply optical mapping to say, if we have molecules that kind of span that region, can we better tease out the structure that's occurring and kind of give us evidence that these 2 genomic fragments are connected in the orientation that we believe them to be connected in. And so again, this is showing that same structure of the dup, trip, dup, and showing that exact same structure that we visualize in our optical mapping data, showing the duplication, triplication, and duplication. And so when we start to kind of tease out that optical mapping data, we have our here structure at the top, and I kind of placed arrows to show where the duplication is occurring, the triplication, and the duplication. And so we can see here, I'm kind of highlighting junction 2 first. So that's the junction that occurs with a unique sequence, and that's a junction that we can visualize in our optical or in our whole genome sequencing and other data pretty clearly, because again, it occurs with a unique sequence. But junction 1 was really the one that we had problems with because it occurs within those repetitive elements that we have no information from our whole genome sequencing data. But really nicely, when we kind of zoom into that region, we can really understand how those 2 duplications and triplications are connected together. And so here at the very top, this is again showing our optical mapping data. At the very top, I've just highlighted where the genes MECP2 are and where that opsin cluster, so those gene repeats that I had talked about earlier. And we can see here effectively a visualization of that junction 1. And so that's basically the junction that's the end of that second duplication connecting to the end of the triplication. And what's nice is that we actually have single molecules that span throughout that entire junctional event. And so normally we would be really limited by our ability to connect those 2 pieces together because of those repetitive elements that kind of split the regions apart. But since we have single molecules through optical mapping that span from one side to the other, we can actually see through the entire region. And again, we can see through it in cis, so in the single DNA molecule, connecting those 2 genomic fragments together. And so again, I kind of highlighted here our junction 2 that we can see in optical mapping as well in whole genome sequencing data. But our junction 1 we can only visualize in our optical mapping data because of the repetitive elements of where those 2 junctions or where those 2 genomic fragments are connecting back together. And so really interestingly is not only do we have that structure, but we can also start to tell finer details that weren't apparent before. And so specifically here, we can really see at that junction what genomic fragment is actually connecting to what other genomic fragment. And so when we really look at that junction 1 closer, we can actually tell that it's not just the triplication that seems inverted, but it also seems that the duplication is in an inverted orientation as well. And so when we go back to a kind of newly refined structure, so we started out with this duplication, triplication, inverted duplication structure. But when we kind of really look at the optical mapping data and try to see where those sequence motifs are lining up, we can actually see that it's not just that triplication that's inverted, but it also appears that the duplication itself is inverted. And so I think really importantly, that doesn't actually change what our junctions are, right? So junction 1 still connects the end of this duplication with the end of this triplication. And junction 2 connects the beginning of the triplication with the beginning of the duplication. And when we look at our newly refined structure, those junctions themselves are the exact same, but the genomic fragments that form those junctions and their inverted status is what we can refine and kind of better delineate in this sample. And so this is great for one sample, but we kind of want to say, okay, we have a lot of these samples that have this duplication, triplication, duplication structure. Is this a feature that we find in other samples with the same genomic aberration? And so can we visualize this new structure, or this refined structure in other MECP2 duplication samples with that same triplication that's occurring? And so, again, luckily we have a lot of these samples in the laboratory, and they've been very well studied. And so this is just kind of showing Claudia's 2011 Nature Genetics paper that kind of shows a lot of these duplication, triplication, duplication samples, and really refining the structure here. And I just want to highlight this sample that we went through, labeled as BAB2772. And this is one with a fairly large duplication, but then a very small triplication that's occurring. And so if we look at this on an array CGH, we can see again a copy number neutral going up to a duplication, going up to a triplication, down to a duplication, and back down to copy number neutral. And nicely here, we can actually see this is inherited from his mother. And again, mothers or females can carry this MECP2 duplication structures because they basically have another X, versus males, since this is on the X chromosome. If you have this complex structure on your only X, then you are affected. And so kind of what I wanted to show here is basically that same array I kind of lined up in the exact same view. So we have the array up here at the very top showing where our MECP2 gene is, as well as the gene TKTL1, which is within our triplicated region. And then in red here, I just kind of show where that duplication goes. And then a small blue is our triplication. And then we have the red portion here as our duplication. And so what we can kind of do is almost like be detectives. And we can say, okay, I know the sequence motif structure that encompasses that triplication. Where do I see that structure in our optical mapping data? And so what we really do is kind of look in our optical mapping data to say, where do we see that triplication structure occurring? And when we do that, I kind of just have highlighted here. Up here in the green is where our reference is. And what I highlighted in this blue box up in this reference is where that triplication motif is occurring. And then below that, in these blue, I kind of highlighted where we see that motif in all of these different contigs that occur in our optical mapping data. And so if we really zoom into that region, we can see exactly the sequence motif structure that occurs at the triplication, going down to the genes FLNA and EMD, and then kind of going through to a copy number neutral state. And again, this is a region that was very well studied by Claudia in 2011 to try to kind of tease out if this structure is inverted because of the low copy repeats that occur at that point. And so what we can see here is basically that same structure where it's not just the duplicate or the triplication that seems to be inverted, but when we look at the sequence motifs in our optical mapping data, we can see that the duplication also appears to be inverted to form this structure. And again, these are methods that Claudia in 2011 tried to use for southern blotting, but we were kind of limited, or she was limited by what we could really see with that technology. And so using optical mapping and using kind of our ability to see the entire region in a single DNA molecule to really understand what's connecting before that molecule and what's connecting after allows us to kind of tease out the true structure of what's occurring at this locus. And again, here, I think it's always nice. We can visualize kind of the raw molecules that make up that, that call. And we can see that there are molecules that effectively span before this mutation or before this event into the duplication, going right through both of the junctions through the entire event. And so this is really important for us because this gives us evidence that this is happening on a single DNA molecule in the structure that we are proposing that it's occurring in. And so what we're able to do is, again, resolve that final structure to be not just a triplication that's inverted, but also a duplication that's inverted with the triplication, forming that junction 1 and 2 at this genetic loci. And so, I think what's really amazing is that when we look at this structure, this is actually one of the proposed structures that the Pan-Cancer Genomics Consortium said may form this duplication-triplication duplication formation. And so you can see kind of here they had or, because they knew, as well as we knew, that those junctions are occurring, connecting those two fragments together, but they just weren't sure and were limited by the technology in truly understanding what fragment was actually forming the junction and what position that fragment was in. But using optical mapping, we're able to see through single molecules that the junctions are forming with that duplication and triplication both inverted together. So those are samples that we're just now starting to send more samples for optical mapping, or planning to send more samples for optical mapping to say, can we really see this structure in samples that maybe have a bigger triplication or a smaller duplication, to say, is this the true structure, and can we see this in other samples through optical mapping? So that research is really, really exciting, and it's ongoing, and it's been really fun to work on. But we also wanted to say, okay, we have a lot of very complex genomic features at this MECP2 locus. Can we map the architecture better using optical genome mapping to kind of tease out the true structure? And so I just wanted to go through a few examples that have been really, really fun to work out. And so this is showing an array CGH showing a full view of the X chromosome. So we have the centromere here. We show all of the short arm as well as all of the long arm. And you can see at the very end of the long arm, there's a duplication that spans right through the telomere. And so within that duplication is actually MECP2, which is why this individual has MECP2 duplication syndrome. But when we look at the array CGH, we can also see on the P arm, there's actually deletion that's occurring at the P arm going also through the telomere. And so when we look on an array CGH, all we see is that duplication occurring at that point. And so that's kind of what I was saying. One of the limitations of an array CGH is that although it gives us a very precise copy number call, it gives us no information to what that copy number variant is actually connected to. And so that's something that we wanted to start to apply optical mapping to say, can we better kind of tease out the structure in these individuals? And so here I kind of lined up the exact view of where we see on array. And in optical mapping, we can see that there are sequence motifs that are connecting on the other side of where that duplication is actually starting. And so what our proposed structure here is actually to form what's known as a recombinant chromosome, which I'll go over in a second. But effectively here on the very right side, you can see we have that duplication. And at the beginning of the duplication, we can see there are sequence motifs that connect to another genomic region. And so we're proposing that those connections are actually on the P arm or on the other chromosomal arm that's connecting that duplication at the exact point that was deleted on the P arm. And so this is a structure that we know happens when basically the mother has an inversion of her chromosome that occurs. So this is a figure from a paper that we recently had come out in Human Mutation that kind of looks at these inversion states and looks at how recombinant chromosomes can be formed. And so if we start at this very top, we can see if the mother has an inversion that occurs, here highlighting just chromosome 3 as an example, but the mother has an inversion between that C and B. So C and B are basically inverted. And when her chromosomes go through homologous recombination, one of the outcomes that can occur because of that inversion is this recombinant chromosome that has a deletion on the P arm and a duplication on the Q arm. And basically that duplication in reality is actually one fragment is on the Q arm and the other fragment is connected on the P arm. And so this is a consequence of when a mother has an inversion and then has a child, that child can have a recombinant chromosome. And so we have kind of a proposed architecture of how we view this structure in an array CGH, what our proposed view is for our proband, so kind of having those duplications connecting on the P and Q arm, as well as our proposed architecture in the mother. And so this would be that the mother has a pericentric inversion that spans the centromere, basically of her almost full chromosome X. And so when we look in the optical mapping data, we can actually see that that connection, or where those genomic fragments were on the Q arm, actually does connect to those locations on the P arm. And so just to kind of put a pin on this, so that kind of proves to us that this is a recombinant chromosome. And from an array, we saw that duplication deletion structure. And from optical mapping, we could actually map that structure back to prove that this was actually a recombinant chromosome by using our optical mapping data to connect those two genomic loci together. And so kind of just one final example, and this was a really complex one that was so interesting to kind of work through. So this is a duplication-triplication duplication structure that's occurring with what appeared to be a deletion on the Y chromosome that is an apparent translocation. So you can see this was another sample that was highlighted as being studied in Claudia's 2011 Nature Genetics paper with a very large duplication, a fairly large triplication, as well as that duplication. And so if we look at the array CGH, we can see a full view of chromosome X as well as a full view of chromosome Y. And we can see on that chromosome X at the very end of the long arm, we can see that copy number variant of a duplication-triplication duplication structure. And then if we look on chromosome Y, we can see that deletion is occurring at that specific point on the Y chromosome. And so when we see something like this, we suspect this to be a translocation. But again, an array CGH gives us no information to how these genomic fragments are connected together. And so although we propose that presumably this triplication and duplication are somehow connecting to that Y chromosome, from an array CGH, we have no information that these genomic fragments are actually connected in a way that we believe they are. And so I just kind of wanted to zoom in so we have a better view of this array CGH. And again, we have our copy number neutral going up to a duplication, spanning, going then up to a triplication, and then back down to a duplication through that telomere. And so what we did is when we look at this region in our optical mapping, we can start to kind of map where things are. So I think it's good to kind of anchor ourselves to what's occurring. And so what we have here is I kind of just placed MECP2 is occurring within that triplicated region. And we can see there are genomic features in our optical mapping data that something is occurring in this region that we can then start to kind of solve the puzzle. And what I really wanted to visualize is kind of our array CGH on the same kind of size view as our optical mapping data. That's basically showing our array CGH duplication starting in the same location of it starting in our optical mapping, going up to that triplication that's forming the structure in optical mapping, going back down to a duplication. And so I kind of highlighted here these sequence motifs that are connecting to that triplication in a way that is not to reference, right? So we know that triplication is connecting to something else that's not in the reference for that region. So again, just labeling MECP2 being right there. And so if we kind of line up that same view on the Y chromosome, we can see that there's a deletion that's occurring in the array CGH. And if I line up that same view in optical mapping, we can see that at that deleted region, there is actually sequence motifs that are connecting to something other than reference, right? So we know that at that genomic location, those sequence motifs are connecting to whatever side the breakpoint is, right? So connecting to whatever that DNA molecule is fused to. And so when we try to think of the structure here, I kind of labeled out through arrows kind of our level of copy number gain. So remember, this is a male, so we're working on the X chromosome. And so when it's a duplication, we have 2 copies. When it's a triplication, we have 3 copies. And so we see our array CGH, our dup, trip, dup. And so we can kind of start to propose a structure that we believe that the Y chromosome is connecting to that triplication in an inverted orientation, and then the rest of that dup, trip, dup structure continues. But again, this is just a hypothesis, and from the array, we're not actually able to prove this hypothesis exists. And so that's why we wanted to go into our optical mapping data to really see if this structure exists in this fashion that we believe it does. And so what we first do is can see junction 2. So again, junction 2 is basically the beginning of that triplication connecting to the beginning of that duplication. And we know that the triplication should be in an inverted orientation. And so I kind of labeled the arrows here being a red arrow and green arrow are now optical mapping data. And if we look at that green arrow where the triplication occurs, we can kind of zoom into that region of where it is inverted and then connected. And so I kind of zoomed in on this box that we know that the triplication is inverted, connected to the front of that duplication, and which is kind of that bigger box at the very bottom. And so one thing that's noteworthy is that we know through molecule support that those 2 genomic fragments are connected in that orientation. So that gives us proof that that junction exists in the architecture that we believe. One question that we're still kind of working through with this sample is that there also appears to be some genomic material that's connected between those 2 fragments that we haven't yet been able to identify. that we're still working on, but we at least know the structure exists with those two junctions connecting. And so when we move on to the Y chromosome, we can look at those sequence motifs that are basically connecting at the point that the deletion we know occurs on the Y chromosome. And when we kind of take that mapping of that MECP2 region that's triplicated, and we look and kind of connect it to that region on the Y chromosome, we can see that the MECP2 triplication is actually also connecting in an inverted orientation to the Y chromosome when we really look at the sequence motifs and we can match them up together. And so just as a final check, those sequence motifs that were on the X chromosomes, now I kind of have two views from the optical mapping, the Y chromosome at the very top, and the X chromosome we kind of flipped upside down to make it a little bit easier to understand. We can actually see that those sequence motifs are connected from that Y, or from the X chromosome after the triplication, match up exactly to what would be on the Y chromosome at that exact location. And so if we kind of zoom in a little bit here, we can see that the sequence motifs are matching up, connecting those two genomic fragments together in the orientation that we would propose here. And so what we bring together is finally we can actually resolve this structure. So our hypothesis that it was the Y chromosome connecting to that triplication that was in an inverted orientation, then connecting to the rest of the structure, we now have evidence through optical mapping data that those two junctions occur within the region that we would propose them to be. And so just some kind of final conclusions to kind of look at all of this optical mapping data together. Really, I think the visualization of long unbroken DNA molecules is really critical in truly understanding a structural variant. And I think methods like optical genome mapping has really helped us kind of tease out structures in ways that weren't possible before. And again, as I was kind of mentioning through the talk, is that looking at these structural events in cis, and that is in a single DNA molecule, is really critical in teasing out the true structure. So if we think of other methods, we're shredding the DNA, we're kind of trying to paste it back together in a way that we think, and it's much more difficult to us to understand an event in its totality. And again, I kind of wanted to highlight the severity of MECP2 duplication patients. So we're just now starting to understand how complexities can lead to more severe MECP2 duplication patients. And so what I think is noteworthy is that for a long time, they had no ability to visualize that some portions of MECP2 were actually triplicated. And so as arrays become higher resolution and they could see where triplications were occurring, they actually found that MECP2 patients who had a triplication of MECP2 actually had a more severe phenotype. And so I think we're just now starting to understand, now that we can really piece these complexities back in the MECP2 locus together to find their true structure, we're just now looking at that genotype-phenotype correlation to better understand if these patients with really complex structures, and again, now that we know the true structure, if they have a more severe phenotype than those who have just a simple structure at this locus. And kind of finally, as the roles of structural variants and kind of their role in gene disruption and dosage expression of pathogenic alleles, and kind of truly understanding how those lead to human disease and how those lead to disease traits is really, really important. And I think methods like optical mapping and other technologies that really help us understand the true structure really let us look at these aberrations in their totality and in a way that just wasn't possible before. And so I just kind of want to end with, I'm sure we've all had a lot of nice reading through the pandemic, but really a really great book was called The Great Influenza that kind of talked through the 1918 Spanish flu and really the history of medicine and the experimental method and the history of science. And I thought a really cool quote in there was from Jacob Heinel, who was one of the fathers of modern germ theory, and he said, "Nature answers only when she is questioned." And for me, it made me think that really methods like optical genome mapping and these new methods that are coming out really allow us to ask new questions. And when asking those new questions, we get new answers, and we can refine structures, and we can really understand what's happening at these genetic locus in a way that wasn't possible before, and we can really push our understanding of these structural variants and their role in disease forward. And with that, I just want to thank my mentors, Claudia Carvalho and James Lupsky. They're really such amazing mentors, and it's been so fun to work on these projects, as well as our collaborators at the Karolinska Institute and the University of Sao Paulo, and our generous funding sources at the National Institutes of Health, as well as Baylor College of Medicine and Bionano for a lot of their technical help and really helping us with some of these very complex cases. And with that, I can take any questions.
Chris, can you hear me?
Yes, I can.
Fantastic. Oh man, you have such an amazing talent for making this extremely complex content digestible for our viewers. We have a lot of people writing in wanting a copy of your deck. I imagine they want to take a little bit of time on their own to review the slides. But just so everybody knows, this broadcast will be available for a few months after we close today. Chris, I think I can condense a lot of the questions that have rolled in into this one before we get to the specific questions on our list. What specific technology, and you may have mentioned a few in your talk, but what specific technology or combination of technologies were you using and for how long before, and how long have you been using optical genome mapping in your research?
Yeah, so we've been using kind of optical mapping for about a year or two to kind of work through these complex samples. And again, Dr. Lupsey's laboratory and Claudia Carvalho has really been working on these structural variants for many, many years and decades. And I think they've used, and again, I showed in the talk kind of southern blotting that was used, arrays, whole genome sequencing. So I think it's like each time a technology is like pushed forward or our understanding, then we like know a little bit more and we can ask kind of a new question. And so it's like when arrays really started to get high resolution, we saw things we didn't see before. When whole genome sequencing allowed us to see every single nucleotide, we saw things we didn't see before. And I think optical mapping is, again, one of those technologies that now it allows us to ask these new questions. And so I think it's like we can constantly build on the technologies that are available to ask new questions. And even with the chromosome 6 inversion case, it's like, if you just stopped at the karyotyping, you would have like solved the phenotype. But it's like, as you apply new methods and you apply higher resolution, you can actually see that there are more complexities that weren't appreciated previously.
Okay, so I don't know who the author is, but it kind of makes me think of that quote, the more you learn, the less you know. Sort of
it becomes like a rabbit hole where it's like, oh, so it's actually like that. And oh, it's like, as we can see more, it just gets more and more complex. And so you're solving things, but then it also leads to new questions. Right, right.
All right, well, let me start with this first question from Julie in Montreal. And she asks, does this technology work on DNA extracted from fresh blood, or can it work on DNA extracted from frozen blood?
Yeah, so that's a really good question. So a lot of our samples are coming from cell lines. So we have basically lymphoblastoid cell lines that have been frozen down over the years that we've then sent for optical mapping, but it can be done on fresh blood as well as frozen blood. And as far as I understand, there are some reagents that you can put into the blood before it's frozen down to keep it more stable, because then you really need that high molecular weight DNA. And so that's why we kind of need either fresh or frozen blood or cells that have been frozen down to capture that DNA. Because if the DNA was just extracted through normal methods, then it wouldn't have molecules that are long enough. So, yes.
Okay. Yes. All right. Next question comes from Lemuel in Alberta. Have you identified key clinical features associated with the types of genomic complexities observed in the MECP2 story?
Yeah, so that's a really good question, and that's actually what we're working on right now. And so, again, I had kind of told the story of the triplications, that we know that those cause a more severe phenotype, but we don't really know yet if complexities lead to a more severe phenotype. And so kind of the genesis of really going through these MECP2 samples are to solve the true structure. And then once we solve the true structure, we can go back and look at the clinical features of each of those patients and really say, was this a more severe patient that had a more complex genomic presentation?
Got it. Okay. Meredith is curious, have you made any comparisons between NGS and optical genome mapping on the samples back and forth?
Yeah, so it's definitely things like the ARID1B and the multiple de novo on chromosome 6. So that was one that we kind of looked together at the NGS and optical mapping data together. And for us, things like the MECP2 duplication syndrome, that's a cohort where NGS really has trouble because, again, we're looking at those repetitive regions that if we just do normal whole genome sequencing, the breakpoints are occurring at regions that whole genome sequencing is not capturing. And so for some cases, we are looking at them both together, but for other cases, we know NGS is just not effective for us to analyze.
Understood. And this kind of piggybacks onto that from Ben, UCSD. Can you describe your experience trying to detect inversions genome-wide with other methods? Or is it go back to that new technologies coming to the forefront?
Totally. So that's a really good question. And Claudia Carvalho, now at the Pacific Northwest Research Institute, her lab is really dedicated to truly understanding and kind of identifying these inversions genome-wide. So optical mapping is a technology that we're trying to use to really see how well can we detect inversions that may have been missed by other methods or may have been not properly called by other methods. So that is an ongoing research project and aim to kind of look genome-wide to say, are there inversions that, because they were just occurring within repetitive regions, that may have been missed or may be in the reference, just not seen? And so using optical mapping, we're kind of able to better see inversions that may have been invisible genome-wide by other methods.
I'm guessing your bandwidth doesn't allow you to be that exact person to be comparing all of those.
No, but Claudia has an amazing new lab, and she's really gearing up to do very amazing things with inversions.
All right, okay. Let's see, Jacqueline. You mentioned ultra-high weight molecular DNA. What can you tell us about the sample preparation process before a sample is run through the Bionano?
Yeah, so that's a really good question. And I would kind of, to better point to, so Bionano has basically extraction procedures. And so there are very kind of precise things that you want to do to make sure you preserve that ultra-high molecular weight DNA. And so the sample preparation involves special reagents that basically preserve that high molecular weight. So for the more technical aspect, I would definitely point you to kind of Bionano's protocols for extraction.
Gotcha. All right, I think we are at the top of the hour, Chris. Any last or final words?
I think, yeah, thank you for the opportunity. It's always fun to kind of present our research to everyone and kind of condense our thoughts together to a coherent story. So I hope I did that.
No, you did a great job, and many thanks to you. This was these slides are amazing, and it must have taken a great deal of time. And we're really thankful to you and to the Lupsky Lab and everybody working on figuring all of this out. So thank you so much for your time today.
Definitely, thank you.
All right, everyone. Thank you for joining us. Have a wonderful rest of your week, and we'll talk soon.